A History of Microbiology. Miasmatic theory. The miasmatic theory postulates that disease caused by bad air or malaria, known as miasmatic odors. It was thought that these miasmatic odors arose from decaying organic matter and was the cause of diseases like cholera, chlamydia, and the bubonic plague. Although incorrect, the miasmatic theory spurred the development of better sanitation and hygiene practices. Aqueducts were built that brought in fresh water. Sewers were built that carried away waste and sewage. This practice protected the Romans and from many waterborne diseases. Hippocrates was coined the father of Western medicine. The Hippocratic Oath is also known as the physician's oath. It is the oath that each physician takes before becoming a medical practitioner. It is an oath to uphold ethical standards and to refrain from judgment or bias and to do no harm. Spontaneous generation is the theory that living things can come from non-living things. This idea is actually so old that we are unable to trace where this theory began. As odd as this idea may seem, we need to remember that they only knew what they knew. They made observations, and they came to conclusions based on those observations. Not only was this idea somewhat universal, but it actually took centuries to convince even some of the most creative scientific minds of the fallacy or untrue nature of the theory of spontaneous generation. So, what were the observations that led to the theory of spontaneous generation? Without microscopes, it did appear as though animals or insects were spontaneously appearing out of non-living things. For example, fleas might appear out of dust. Maggots would seemingly appear from rotting meat. Mice would come out of a pile of corn husks. And flies would seemingly generate from a pile of manure. The pneuma was theorized as the spiritual essence or vital heat that non-living material must possess in order to undergo spontaneous generation. In the 1600s, two scientists on opposite sides of the theory of spontaneous generation battled it out in the lab. Antony von Leeuwenhoek and Francesco Redi. In 1668, Francesco Redi did the following experiment. Redi performed an experiment using jars containing rotting meat. One jar was covered with gauze to prevent flies from coming into direct contact with the meat, while the other jar was left open, allowing flies to come into direct contact with the meat. One of the ideas of spontaneous generation was this pneuma or vital force, which was believed to be the spiritual force that led to spontaneous generation. The pneuma was believed to travel through the air. This is why the jars were covered with gauze and not simply closed tight with lids. So question 1 is, do maggots spontaneously generate from meat, or do maggots come from flies coming into contact with meat? So we set up the experiment. We allowed meat to sit out unprotected from flies and protected from flies. If no maggots were observed in meat protected from flies, then the question number 2 must be correct. Maggots come from flies that must come into direct contact with the meat. Possible outcomes. If spontaneous generation was correct, then both samples of meat should produce maggots because they are both exposed to the air. If spontaneous generation was incorrect, the meat inside the covered jar will not produce maggots. If flies produce maggots, then maggots will appear in meat only after there has been direct contact with flies. Outcomes. The meat inside the uncovered jar was exposed to direct contact with flies and produced maggots. The meat inside of the covered jar that was exposed to the air only, or the pneuma, but protected from flies, did not produce maggots. This was the first evidence against spontaneous generation. His experiments also demonstrated that flies must come into direct contact with the meat in order for maggots to appear. The discovery of cells and the discovery and the development of cell theory was largely due to the invention of high-powered microscopes in the 17th century. Robert Hooke discovered cells in 1665 by looking at a piece of cork under a microscope. Cork is made up of dead plant cells. He only recognized plants as having cells, but not animals. Hooke named the structures he saw in cork cells, after the cells of a honeycomb. Vaccines. In 1796, Edward Jenner, who was working with smallpox, discovered the principle of vaccination. Vaccinations can prevent disease by exposing the subject to a milder form of the disease-causing agent. Edward Jenner observed that milkmaids who had previously been infected with a similar disease called cowpox were protected from smallpox. The principle behind vaccinations is that a disease can be prevented by exposing the subject to a milder form of the disease-causing agent. Interestingly, the term vaccine comes from the term variolae vaccinae, which means smallpox of the cow. Antony van Leeuwenhoek. Early microscopes did not have much magnifying power until 1675, when Dutch cloth merchant named Antony van Leeuwenhoek developed the first microscopic lenses that were powerful enough to view microorganisms. This improvement resulted in a magnification power of up to 300x. Anton van Leeuwenhoek was coined the father of microbiology. He was the first to observe motile microscopic life forms. He called them animalcules, and he observed them swimming in a drop of pond water. Louis Pasteur. Louis Pasteur performed a series of experiments that presented overwhelming evidence against the theory of spontaneous generation. Pasteur designed special flasks containing swan-necked tubes that protruded from the top. This design allowed for air, or the pneuma, to pass through into the broth, but it prevented any microorganisms from passing through the air into the broth. His experiments demonstrated that exposure to air was not enough to grow microorganisms in broth. The broth had to be exposed to microorganisms in the air. Here we can see the general setup. Here we can see the general setup. So first the broth was boiled to make sure that there were no microbes existing in the broth at the beginning of the experiment. Then the swan-necked flask was allowed to incubate. Some were intact, only allowing air to pass through, or pneuma to pass through, but microbes would not be able to enter. In other examples, the swan-necked flask would be removed, allowing both air and any airborne microbes to enter the broth. After several repeats of this experiment, it was found that exposure to air alone, or pneuma alone, was not sufficient to grow microbes in broth. Pasteur also invented a fermentation process called pasteurization. We still use this process today in many of our pasteurized foods, such as some dairy products and our milk. Disease transmission. In the 1700s, people thought that diseases resulted from the foul qualities or rotting particles called miasma in the environment or air. In the late 1800s, careful scientific inquiry into the nature of disease gave rise to the field of epidemiology. Epidemiology is a study of the source, cause, and mode of transmission of disease. John Snow was coined the father of epidemiology. In 1854, John Snow determined the cause of cholera transmission in London. He found that it was due to a contaminated well. This was the first epidemiological study. Germ theory. Louis Pasteur proposes that germs are the cause of infectious diseases, and he popularizes this theory in 1862. The germ theory is that diseases come from germs, not miasmatic odors. The golden age of microbiology began in 1857, largely with Ignaz Philipp Semmelweis. Semmelweis was instrumental in the development of aseptic techniques as a defense against the germs that were the cause of disease according to germ theory, which had not yet gained popularity at that time. Semmelweis discovered that hand washing in obstetrical clinics drastically reduced the incidence of what's called childhood fever. Semmelweis's discovery was tested and proved over and over again. However, the scientists of the day were reluctant to accept that hand washing reduced mortality rates because the idea of miasmatic theory was prevalent and widely accepted within the medical and scientific communities. In fact, many medical practitioners refused to wash their hands and were insulted at the idea that their hands may be contaminated. Semmelweis was coined the savior of mothers. Puerperal fever or childhood fever was common in the 1850s and was often fatal. In fact, 50% of postoperative patients would die due to postoperative infection. Semmelweis concluded that these infections were due to unseen microorganisms that were able to travel through the air or by a contaminated object coming into direct contact with the patients. Unfortunately, Semmelweis's ideas were not popularized until after his death, when Lister and Pasteur continued to work in antisepsis. Louis Pasteur invented a process that reduced the number of pathogens in certain food products, such as dairy products. This process was named pasteurization, and is where we get milk products like pasteurized milk. The process of pasteurization kills most of the bacteria that causes spoilage, thereby increasing food quality and protecting public health. Louis Pasteur also proved that the fermentation process was caused by the single-celled organisms called yeast. John Lister also furthered the earlier work by Semmelweis, who had developed the practice of sterile techniques and demonstrated the link between non-sterile environments and diseases. In 1865, Joseph Lister developed the practice of antisepsis, which is the chemical disinfection of external living surfaces. Lister and Listerine. The medical field adopted many of the aseptic techniques set forth by Lister. These methods of sterility and decontamination became the new standard for patient care. A great many of the aseptic techniques developed by Joseph Lister are still used in the medical field today. Fun fact, Listerine was developed in 1879, but not by Lister. It was developed by Joseph Lawrence and was named after Joseph Lister in honor of his contribution to antiseptic surgery. Robert Koch is known as the founder of bacteriology. Robert Koch performed an experiment that provided evidence that bacteria cause disease. Specifically, Koch identified the pathogens that caused tuberculosis, cholera, and anthrax. Koch experimentally identified four generalized principles linking pathogens to disease, now known as Koch's postulates. Koch's postulates are as follows. Number 1. The organism must always be present in every case of the disease. Number 2. The organism must be isolated from a host containing the disease and grown in pure culture. Number 3. Samples of the organism taken from pure culture must cause the same disease when inoculated into a healthy susceptible animal in a laboratory. And number 4, the organism must be isolated from the inoculated animal and must be identified as the same original organism that was first isolated from the originally diseased host. Dmitry Ivanovsky and Martinus Viziernik. In 1892, Dmitry Ivanovsky filtered the sap from a diseased tobacco plant and discovered that the filtrate was able to infect a healthy tobacco plant with the disease. Independently, Martinus Viziernik discovered the same virus while conducting similar experiments in 1899. Viziernik described this filtrate as a contagious living liquid that acted like a poison or virus. The word virus comes from the word for poison. This is how viruses got their name and how the field of virology was born. Properties of prokaryotes. Important properties of prokaryotic organisms are that they can rapidly reproduce using binary fission. Some species are able to survive harsh conditions. Some are even able to produce endospores, which allows them to survive through long periods of unfavorable environmental conditions. Bacteria have been discovered in some of the most inhospitable environments on Earth. Soil samples, sulfurous underwater sea vents, glacial ice, acidic hot springs, and radioactive waste. They are by far the most numerous life forms on Earth. Pathogens are organisms that cause illness or disease. There are pathogenic organisms that are in the bacteria domain and the eukarya domain, but so far we have not discovered any pathogenic archaea. There are different shapes of bacteria: bacillus or rod-shaped, sphere-like or coccus, spirillum, which is somewhat spiral-shaped. The rod-shaped cells are called bacilli. A cell with a rod shape is called bacillus or bacilli when plural. Most rods occur either by themselves or in pairs. A pair would be a diplobacillus. They can also be arranged into long chains called streptobacillus. Cocci are round-shaped bacteria. A round-shaped bacterial cell can be called a coccus by itself or cocci for plural. This word cocci comes from the word meaning berry, which makes sense due to its shape. They do not have to be perfectly spherical. They can be oval or even a little lopsided. They usually measure only about 0.5 to 1 micrometer. Many cocci arrange themselves in pairs, called a diplococcus. Cocci can also form a cube-like tetrad, which is composed of 4 cocci. An organism that causes gonorrhea, called Neisseria gonorrhea, and one type of bacterial meningitis are both diplococci. When cocci form an 8 cocci cubed, this is called a sarcina, which means bundle. Cocci that form a chain are called streptococcus. Some well-known examples of streptococcus are Streptococcus pyogenes, which causes strep throat, Streptococcus mutans, which causes tooth decay, and Streptococcus lactis, which is harmless and used in dairy products like yogurt. Staphylococcus. Sometimes cocci just cluster together in a random non-cubed formation. This is called staphylococcus. Staphylo means cluster. A well-known example of Staphylococcus is Staphylococcus aureus. Staphylococcus aureus can be the cause of some types of food poisoning, toxic shock syndrome, and several skin infections. You probably better know this as staph. The vibrio is a curved rod shape that resembles a comma. The cholera-causing pathogen Vibrio cholerae is a great example of a vibrio bacteria. Spirillum has a helical shape with a thick, rigid cell wall and a flagella for motility. They are gram-negative. We also have spirochetes. These spiral-shaped cells, called spirochetes, have thin, flexible cell walls and are more mobile. Spirochetes contain a diderm, which means a double membrane. Spirochetes stain gram-negative and are chemoheterotrophic. Spirochetes are distinguished from other bacterial phyla by the location of their flagella, and sometimes this flagella is called an axial filament, which runs between the bacterial inner membrane and outer membrane in the periplasmic space. These cause a twisting motion which allows the spirochete to move about. Archaea have more morphological diversity than bacteria. Some very strange, rather odd shapes are found in archaeal cells, like branched, square, lobed coccus, short or elongated bacilli, or even a coccus with a bunch of flagella coming out of only one side. There are many species of bacteria that act as parasites to humans and other organisms. These parasitic bacteria are called pathogenic because they cause disease. However, there are no known pathogens for archaea. But bacteria are not all bad. In fact, we have very helpful bacteria, or mutualistic bacteria. Bacteria are an integral and even necessary part of the human body. It is estimated that the human body itself consists of 10 times more bacteria cells than human cells. Very few bacteria live independently. In fact, 99% of bacteria live as part of a larger entity called a biofilm. Biofilms display many of the characteristics of multicellular organisms. These cells communicate to each other through chemical signaling called quorum sensing. Cells in a biofilm become physically attached to each other and to a surface using interlacing polymers that they themselves secrete. Here is an illustration of the biofilm life cycle. In stage 1, we have the initial attachment. In stage 1, a free-floating bacteria reversibly attaches itself to a surface. Stage 2 is irreversible attachment. In stage 2, the small colony irreversibly anchors itself to the surface through the expression of cell adhesion molecules and polysaccharides that they will secrete. Stage 3 is maturation 1. The adhesion sites get more complex and diverse, and cell division begins along with cell recruitment. Stage 4 is maturation 2. Here, the biofilm has reached full maturity. The matrix envelops the biofilm and protects and provides protection from harmful elements, including from antibiotics. Stage 5 is dispersion. Occasionally, single cells will be released from the biofilm during cell division. These cells will be dispersed and will be able to travel to form a new biofilm. Quorum sensing. Bacteria cells function very differently when they are together with other cells. The bacteria cells can sense, hence quorum sensing, the density of the population that they are a part of. As this population density increases, the bacteria cells respond by changing their gene expression. For example, bacteria cells that are part of a high-density population, such as a biofilm, will change their phenotype to fit whatever the needs of the biofilm as a whole are at the time. It was not until the invention of the electron microscope that it became clear that bacteria were indeed cells. Due to their size, plant and animal cells were already known at the time. But since bacteria cells are on average about 10 times smaller than plant and animal cells, it took this leap forward in technology to visualize these tiny life forms in greater detail. Tiny eukaryotic cells were also able to be examined in more detail using the electron microscope, including the algae, fungi, and protozoa. Once we could examine smaller cells microscopically, new categories arose due to fundamental differences between these bacteria cells and the eukaryotic cells. Cells can be separated into two categories: prokaryotic cells and eukaryotic cells. Prokaryotic cells do not have a nucleus, and they do not have membrane-bound organelles. Archaea have the same general appearance as bacteria, but upon further examination, archaeal cells have more in common with eukaryotic cells than bacteria cells. Despite their differences, all of the organisms in both the domain bacteria and the domain archaea are entirely made up of what are considered prokaryotic organisms. Eukaryotic cells are compartmentalized. They have a nucleus, which is separated from the cytoplasm by a nuclear membrane or a nuclear envelope. Eukaryotic cells also contain many membrane-bound organelles. Bacteria do not have a nucleus, and they do not have membrane-bound organelles. Also, archaeal cells do not have a membrane or membrane-bound organelles. All of the membrane organelles that eukaryotes have will not be found in prokaryotic cells. This includes the smooth and rough endoplasmic reticulum, or reticuli, the lysosome, the Golgi body, and of course many more. In prokaryotic cells, the cytoplasm would be referring to all of the intracellular contents. The cytoplasm represents everything inside eukaryotic cells that are internal to the cell membrane or plasma membrane, but external to the nucleus. The internal environment of the cell is referred to as the cytoplasm. The cytoplasm includes the intracellular fluid, dissolved substances, and any organelles of the cell, but does not include the nucleus if it has one, or any contents of this nucleus. The inside of the cell is an aqueous environment full of intracellular fluid that is more appropriately referred to as the cytosol. If you were to remove all of the cellular structures from the cytoplasm, you would be left with the cytosol. The cell envelope comprises the cell membrane and any structures that are surrounding the cell membrane. Gram-negative bacteria have a cell envelope that consists of the inner cytoplasmic membrane, the periplasmic space, the outer membrane, a cell wall, and a capsule. Gram-positive bacteria cells have an envelope that is consisting of one singular cytoplasmic membrane and a larger cell wall. Occasionally, depending on the species, you will also get an S layer and perhaps a capsule as well. Most bacteria live in aqueous environments and have the added protection of the cell wall. The cell wall lies on the outside of the cell membrane and gives additional protection. The cell wall protects from osmotic pressure. When a cell wall is placed in a hypotonic solution, the water molecules rush into the cell, but the cell wall resists that added pressure, protecting the cell from lysis or bursting open. Cells that are placed in a hypotonic solution that do not have a cell wall undergo lysis and die. Let's look at the cell wall of the bacteria. The majority of bacteria contain cell walls. The cell walls of bacteria will always contain some amount, either thick or thin, of peptidoglycan. This peptidoglycan is the primary target for many antimicrobial and antibacterial drugs. Archaea also often have a cell wall. Their cell walls are semi-rigid or flexible. They contain no peptidoglycans. Therefore, since they don't contain peptidoglycans, they would be insensitive to the antibiotics that target the bacterial cell wall. Methanogens do have a similar substance in their cell walls called pseudopeptidoglycans. Sometimes instead of peptidoglycans, we can call it murine. Many eukarya species also have cell walls. Cell walls can be found in algae, fungi, and plants. Cell walls of plants contain cellulose, but no peptidoglycan or pseudopeptidoglycan. Cell walls of fungi contain chitin, but also no peptidoglycan or pseudopeptidoglycan. The glycocalyx is an outer layer that is external to the cell wall. The glycocalyx can take on the form of either a capsule or a slime layer. When the glycocalyx layer is thick and covalently bonded to the cell, we call it a capsule. When the glycocalyx layer is thin and loosely attached, it is referred to as a slime layer. The functions of the glycocalyx include protection from desiccation or drying out, evading immune defenses, and adhesion sites for attachment to the host cells, tissues, or surfaces. The slime layer is an external layer of extracellular polysaccharides. This extracellular material is secreted when bacteria become a biofilm. This is a diagram of a gram-positive bacterium. The cell wall comprises a cell membrane, seen here in light brown, and a thick peptidoglycan-containing cell wall, which is the purple layer. It has no outer lipid membrane. The red layer is known as the capsule. The capsule is a polysaccharide outer coating of the bacterial surface and often plays a role in preventing phagocytosis. Some bacteria have pili. Pili are protein fibers extending from the cell surface, found in many gram-negative bacteria. Pili contain adhesion molecules that enable them to form attachments to surfaces for forming a biofilm. Pathogenic bacteria also use adhesins to sense specific target cells and then attach to those cells. Some microscopic organisms in all three of the domains of life have one or more flagellum, or flagella. Flagella are long appendages extending from the cell surfaces. The function of a flagellum is to provide motility. The spirochetes win the award for the strangest flagellum. Spirochetes have a special flagellum which is internal, and it is existing in the periplasmic space. Bacterial DNA is in the form of one single circular chromosome. Also, archaeal DNA is also in the form of a single circular chromosome. In contrast, eukarya DNA is in the form of multiple linear chromosomes, and it is located in the nucleus. Bacterial DNA is located in the nucleoid region. Archaeal DNA is also located in the nucleoid region. In contrast, eukarya DNA is located in the nucleus. Histones. DNA in eukaryotic cells and archaea cells are associated with histone proteins that act to package the DNA into bundles. These bundles are called chromatin. This bundling process is referred to as DNA remodeling. Histones function to package or remodel DNA. Histone proteins are found in eukaryotic cells and archaeal cells, but histone proteins are not found in bacterial cells. Bacteria does not contain introns, but archaea and eukarya do have introns. These introns, you might recall, are non-coding regions in the RNA transcript. Many bacteria cells carry a small circular extrachromosomal segment of double-stranded DNA called a plasmid. Plasmids carry non-essential genetic information, like antibiotic resistance or environmental tolerances. Plasmids can even be transferred from one bacteria cell to another through a process called conjugation. Both bacteria and archaea cells have plasmids. Well, they can have plasmids, but not all of them do. However, eukaryotic cells will never contain plasmids. Interestingly, plasmids do not carry any genes that are essential for normal life processes. Plasmids do alter the sensitivities or abilities of the bacteria that possesses them. These are considered non-essential genes and are classified as accessory genes. For example, some plasmids carry genes that allow for antibiotic resistance, habitat tolerance, and bacterial conjugation, which allows for the exchange of genetic material between bacterial cells. The structure of the cell membrane in bacteria cells and eukaryotic cells is very similar. It is a phospholipid bilayer and has a unique structure. It is made up of an inner layer and outer layer of phospholipids that are oriented with their hydrophobic tails facing one another. Phospholipids are considered amphiphilic because they contain a polar hydrophilic head and consists of two nonpolar hydrophobic fatty acid chains as tails. Anything outside of the cell is considered extracellular, and the contents inside the cell are considered intracellular. The cell membrane protects the cell by creating a barrier between what is inside and what is outside. The fatty acid tails orient themselves towards each other to get away from the water. This provides a barrier between the inside and outside. The cell membrane in eukaryotic cells and bacteria cells is made up of a double layer of phospholipids. Here you can see the structure of the phospholipids for the eukaryotic cells and bacteria cells. You'll see that it is made up of unbranched fatty acid chains, which are connected to the phosphate head using an ester bond. So the type of fatty acids that are found in phospholipids of bacteria and eukaryotic cells are unbranched fatty acid chains that consist of hydrocarbons. The phosphate groups of the phospholipids in bacteria and eukaryotic cells are bonded to the fatty acid chains using ester bonds. However, the structure of archaeal membranes is different. The type of fatty acids found in the phospholipids of archaeal cells are composed of branched isoprene chains. The phosphate groups of phospholipids in bacteria and eukaryotic cells are bonded to the fatty acid chains using ether bonds instead of ester bonds. Archaeal membranes can be composed of either a phospholipid bilayer or a monolayer, or even a mixture of both. Hyperthermophilic archaea will have cell membranes that are composed of only one layer of phospholipids. Other archaeal cells that live in more normal conditions tend to have bilayered membranes. And archaeal cells that live in only moderately extreme environments can have a mixture between monolayered and bilayered membrane. All living organisms have to obtain and metabolize energy to sustain life. There are two basic ways in which organisms accomplish this task. Cellular respiration, which is the process of converting chemical energy into cellular energy, or biologically useful energy like ATP. Photosynthesis involves the process of converting light energy into chemical energy. All cells and organisms require energy to live. The mechanisms by which that energy is obtained and processed varies in different life forms. One part of energy processing that is common or conserved among all life forms is called cellular respiration. All cells, including bacterial cells, archaeal cells, and eukaryotic cells, carry out this chain of energy-producing chemical reactions referred to collectively as cellular respiration. In eukaryotic cells, including eukaryotic microorganisms, this process of cellular respiration occurs in the mitochondria. The mitochondria is a membrane-bound cytoplasmic organelle and is only found in eukaryotic cells. Prokaryotic cells, which include bacteria and archaeal cells, do not have a mitochondria, so the energy conservation process of cellular respiration will take place in the cytosol and the cell membrane in the prokaryotic cell. Mitochondria are thought to have evolved from an ancient bacteria according to the endosymbiotic theory. The way in which the cell respiration occurs in the mitochondria is remarkably similar to the way in which most bacteria cells undergo the process of cellular respiration. This process takes place in the cytosol and the membrane of the bacterial cells, just as it would in the mitochondria. While all living things must undergo cellular respiration, not all organisms are able to photosynthesize. There are photosynthetic organisms that belong to all three of the domains of life. Some of the eukaryotic organisms that photosynthesize, including plants, some of the eukaryotic organisms that use photosynthesis include plants and algae, which is a type of protist. In eukaryotic organisms, the process of photosynthesis takes place in special membrane-bound organelles called chloroplasts. The chloroplasts contain a photosensitive pigment that captures sunlight's energy and starts the process of photosynthesis. Some prokaryotic organisms also undergo photosynthesis. However, they do this without the assistance of structures like chloroplasts. Instead, the processes needed to capture light energy take place in the cytoplasm and in the membrane of the bacteria. Mitochondria and chloroplasts are thought to have evolved as the result of being engulfed within a larger cell. The endosymbiotic theory is defined as the theory that mitochondria and chloroplasts evolved through the symbiotic relationship between a prokaryotic cell and the larger eukaryotic cell that engulfed it. Here is some evidence to support the endosymbiotic theory. Mitochondrial and chloroplast DNA is in the form of a circular plasmid, which is very similar to the DNA of bacterial cells. Mitochondria and chloroplasts are able to reproduce via binary fission on their own within the host cell. Mitochondria and chloroplasts are surrounded by two lipid bilayers, which may have formed as the result of the larger host cell engulfing the smaller prokaryotic cell. Ribosomes. The main function of a ribosome is to be the site of protein synthesis. Protein synthesis, because prokaryotic cells lack compartmentalization, the chemical reactions of their metabolism and processes of DNA replication and protein synthesis all must take place in the cytoplasm. Both eukaryotic cells and prokaryotic cells contain ribosomes. The ribosomes perform the same function in both types of cells, which is to be the site of protein synthesis. Ribosomes consist of two subunits. However, there are structural differences between the ribosomes found in prokaryotes versus ribosomes in eukaryotes. Prokaryotic ribosomes are composed of a 30S smaller subunit and a 50S larger subunit, which combines to make a 70S prokaryotic ribosome. The eukaryotic ribosomes are composed of a 40S smaller subunit and a 60S large subunit, which makes a combined 80S prokaryotic ribosome. I'm sorry, that should say eukaryotic ribosome. Diversity of bacteria and archaea. Before the invention of high-powered light microscopes and electron microscopes, cells were generally classified as either plant cells or animal cells, because that was about all that could be observed. Powerful light microscopes revealed that some cells have no nucleus and no membrane-bound organelles. As science and technology continued to advance, smaller cells were discovered. These included protists, fungi, and bacteria. The first electron microscopes were invented in 1940s. The scientific process usually begins with an observation. Light microscopy is limited by the wavelengths of visible light, which are between 400 and 700 nanometers. Electron microscopes form an image of the specimen by using a beam of electrons instead of light. Since electrons have a wavelength of only about 1 nanometer, the electron microscope is able to magnify a specimen up to about 100,000 times. In 1977, evolutionary biologist Carl Woese discovered a new type of cell that existed as a single-celled organism. This new group of organisms were originally called the archaebacteria. Carl Woese made this discovery during his work comparing the nucleotide sequences of different types of cells to uncover the phylogenetic relationships. A campaign was then started to add a new domain dedicated to this newly discovered organisms. The name was later changed from Archaebacteria to Archaea, and the domains were changed to include these new prokaryotic organisms. The domain Archaea was born. So now the phylogenetic tree of taxonomy now categorizes all organisms into 3 domains: bacteria, archaea, and the eukarya, sometimes called eukaryota. Cells can be categorized into 2 broad categories: prokaryotic and eukaryotic. All archaea and bacteria are prokaryotic cells. The eukarya are eukaryotic organisms which are composed of 1 or more eukaryotic cells. The term eukaryotic translates to true nucleus. The term prokaryotic translates to before nucleus. The term archaea translates to the ancient ones. Eukaryotic cells have membrane-bound organelles and DNA, which is in the form of linear chromosomes that are housed within a nucleus that is encased within the nuclear envelope, or nuclear membrane. In contrast, prokaryotic cells do not contain a nucleus or membrane-bound organelles. However, prokaryotic cells do still contain organelles and genetic information. The genetic information contained inside prokaryotic cells is in the form of 1 single circular chromosome, which exists in the cytoplasm. Classifications of prokaryotes. Eukaryotic organisms are made up of 1 or more eukaryotic cells and belong to the eukarya or eukaryota domain. Prokaryotic organisms are composed of a single prokaryotic cell and belong to either the bacteria domain or the archaea domain. The bacteria domain is separated into 5 groups: proteobacteria, chlamydia, spirochetes, cyanobacteria, and gram-positive bacteria. In the phylogenetic taxonomic classification system, the proteobacteria, which is the phylum, is broken down into 5 major groups called classes. Here are the 5 classes of proteobacteria: alpha proteobacteria, beta proteobacteria, gamma proteobacteria, delta proteobacteria, and epsilon proteobacteria. Proteobacteria is a phylum that gets its name from the word proteo, which means first. The Proteobacteria phylum is the largest and most diverse bacterial phylum. Many of the common gram-negative bacteria that microbiologists and medical professionals are familiar with belong to this phylum. Let's discuss the properties of alpha proteobacteria. Alpha proteobacteria are gram-negative. They're also oligotrophic. That means that they can survive in habitats that provide very little nutrition or nutrients. The alpha proteobacteria are also known as extremophiles and live in extreme environments, such as sediments deep in the ocean, or soil deep below the Earth's surface, or even inside of glacial ice. One genus, the Rhizobialis, creates nitrogen through the process of nitrogen fixation. Nitrogen fixation is the process by which the bacteria is able to take atmospheric nitrogen, which is nitrogen gas, and it can turn it into ammonia. The alpha proteobacteria phylum also includes some pathogens. The rickettsias, for example, are pathogens within the alpha proteobacteria phylum that cause Rocky Mountain spotted fever. Rickettsias are obligate intracellular pathogens, which means that they must invade a host cell in order to complete part of their life cycle. The mitochondria in eukaryotic cells were most likely formed from the alpha proteobacteria phylum. Next, let's take a quick look at the beta proteobacteria. The beta proteobacteria are eutrophic or copiotrophs. Copiotrophs or eutrophic bacteria require a lot or copious amounts of organic nutrients in order to survive. Nitrosomonas is a genus of rod-shaped chemoautotrophic bacteria. This organism oxidizes ammonia into nitrite as a metabolic process. These bacteria are important for the nitrogen cycle. Plants depend on the presence of nitrates in the soil in order to grow. The beta proteobacteria includes the bacteria in gonorrhea that causes gonorrhea, and also in meningitis that causes bacterial meningitis. Several species of beta proteobacteria cause illnesses of the respiratory tract, including pertussis or whooping cough. Gamma proteobacteria are highly motile, aerobic, non-fermenting, and they are also known to be resistant to many antibiotics. The Gamma proteobacteria class includes the species P. aeruginosa, which is a highly motile, aerobic, non-fermenting bacterium. Many of these bacteria are symbiotic to humans. However, some of these can cause a number of infections in humans. P. aeruginosa causes infections of the urinary tract and the respiratory tract, as well as open wounds. These bacteria are resilient and resistant to many antibiotics. Gamma proteobacteria also includes the Enterobacteriaceae. The gamma proteobacteria includes these enterobacteriaceae, which are of certain importance because these are often the intestinal bacteria, which are known to be fermenting facultative anaerobes. This family is made up of two subcategories. These are our coliforms and non-coliforms. Coliforms are able to ferment lactose completely into an acid and a gas. An example of a coliform is E. coli and also Vibrio cholera, which is a gram-negative, comma-shaped coliform bacteria that causes cholera. Non-coliforms either cannot ferment lactose or ferment it incompletely into either an acid or a gas, but not both. An example of a non-coliform bacteria is Salmonella. Salmonella can cause food poisoning and typhoid. One type of non-coliform bacteria is the Yersinia pestis, which is known to cause the bubonic plague. The delta proteobacteria are gram-negative bacteria that have the ability to reduce sulfate. This sulfate-reducing bacteria assists in biogeochemical recycling. Also, we have sulfate-reducing bacteria Desulfovibrio orale, which causes gum disease. And last, we have epsilon proteobacteria. The epsilon proteobacteria live in the digestive system of the human body. Epsilon proteobacteria includes many bacteria that find their home in the digestive system of the human body. One of these is H. pylori. H. pylori is the bacteria that is responsible for stomach ulcers. The next phylum we will discuss are the Chlamydia. The bacteria domain also includes the Chlamydia. The Chlamydia phylum includes all the members of the Chlamydia phylum are so small they cannot be seen with light microscopy. Chlamydia is a bacterial phylum and class whose members are non-motile, obligate, intracellular pathogens. They tend to be ovoid in shape and stain gram-negative. Another group of bacteria are the spirochetes. The phylum Spirochetes includes the spirochetes. The bacteria phylum Spirochetes has many spiral gram-negative bacterial species. Some are found in the soil, and others are found in the digestive tract of animals. Spirochetes are distinguished from other bacteria phyla by the location of their flagella, sometimes called axial filaments, which run between the bacterial inner membrane and outer membrane in the periplasmic space. These cause a twisting motion which allows the spirochete to move about. The spirochete phylum includes the following disease-causing species: Treponema pallidum, which causes syphilis, and Borrelia burgdorferi, which causes Lyme disease. Next, we'll talk about the bacteria called cyanobacteria. The Earth formed over 4.5 billion years ago. The early atmosphere was oxygen-free and was composed of carbon dioxide, nitrogen gas, and water vapor.
At some point in our early Earth's history, the Earth cooled enough for water vapor to liquefy, covering the Earth with precious life-giving water. The earliest fossils, the earliest fossil evidence that we have of life on our planet is over 3.5 billion years old. The earliest known life form was a single-celled organism called cyanobacteria. Cyanobacteria use photosynthesis and occasionally chemosynthesis to meet their energy needs. Photosynthesis uses up carbon dioxide from the atmosphere and releases oxygen as a waste product. Photosynthesis converts sunlight's energy, or photons, into biological useful energy, ATP. Over the course of hundreds of millions of years, trillions of these cyanobacteria transformed the early Earth's atmosphere to contain the oxygen we breathe. The addition of oxygen to the atmosphere paved the way for new life forms to evolve that depended on oxygen, like humans do. Cyanobacteria, or cyanophyta, are a phylum of bacteria that get energy through the process of photosynthesis. The cyanobacteria are the only prokaryotic organisms that are able to produce oxygen using photosynthesis. The cyanobacteria perform photosynthesis using the photosensitive pigment chlorophyll that exists in their cytoplasm. Cyanobacteria have chlorophyll in the cytoplasm. They do not have chloroplasts. The chloroplasts of eukaryotic cells probably were the cyanobacteria. According to the endosymbiotic theory, a prokaryote grows in size and develops infoldings in the cell membrane to increase its surface area to volume ratio. The enfoldings eventually pinch off from the cell membrane, forming early endomembrane system. It encloses the nucleoid, making the new membrane-bound nucleus. An anaerobe, or oxygen-using bacterium, then enters the eukaryote, either as prey or perhaps as a parasite. Somehow, this anaerobic bacteria avoids digestion and becomes endosymbiotic. It ends up becoming a cell living inside of another cell. This bacteria aerobe's ability to use oxygen to make energy becomes an asset for the host cell. So the host cell allows it to thrive in an increasingly oxygen-rich environment as the eukaryotes go as the other eukaryotes go extinct. The proteobacterium is eventually assimilated and becomes what we now know of as our mitochondria. In a similar fashion, we believe that some eukaryotic cells probably engulfed cyanobacteria in a similar way. So some eukaryotes go on to acquire additional endosymbionts like the cyanobacteria, which we believe is a group of bacteria that became the chloroplasts. This allows the eukaryote carrying the chloroplasts to undergo photosynthesis. The last group of bacteria that we will discuss are the gram-positive bacteria. Another way that we categorize bacteria is based on how they react to the gram stain procedure. In a nutshell, the gram stain procedure includes a primary stain, which has a purplish blue color. the crystal violet is flooded on top of the slide. The peptidoglycan in the cell walls of bacteria pick up the crystal violet stain. Next, we have a decolorization step. During the decolorization step, the amount of crystal violet taken up by the thin layer of peptidoglycan from gram-negative bacteria is washed away and decolorized completely. Whereas gram-positive bacteria that have larger, thicker, more robust peptidoglycan cell walls that have larger, thicker, more robust peptidoglycan layers within their cell walls will retain the crystal violet stain following the decolorization step. The final step of the gram stain procedure is to counterstain with safranin. The main reason we do this is so that we can visualize the gram-negative bacteria. Otherwise, they can be very difficult to visualize. The addition of safranin colorizes the gram-negative bacteria. It also would colorize the gram-positive bacteria as well, but we find that the crystal violet overpowers the safranin stain. So here are the gram staining results. Here are the gram staining results of different types of bacteria. So we have gram-positive, gram-negative, and here we also have an atypical bacteria, just for fun. So our gram-positive bacteria are going to appear purplish blue because these bacteria retain the crystal violet primary stain even after the decolorization step. The primary stain blocks the pinkish red safranin counterstain from view because the bluish purple is darker and therefore more dominant. Gram-negative bacteria will appear pinkish or red due to the safranin counterstain. They do not retain the bluish purple crystal violet stain after the decolorization step. Atypical bacteria do not have a cell wall. They remain colorless. These bacteria do not retain the crystal violet primary stain or the counterstain safranin. Here is a depiction of a gram-positive bacteria cell wall and a gram-negative bacteria cell wall. Gram-negative bacteria will have a thin layer of peptidoglycan. In contrast, the gram-positive bacteria will have a much thicker peptidoglycan layer within their cell walls. Streptococci are gram-positive aerobic organisms that cause many disorders, including pharyngitis, which is when you lose your voice, pneumonia, wound infections, and skin infections, also sepsis and endocarditis. Staphylococci are also gram-positive aerobic organisms. Staphylococcus aureus is the most pathogenic. It typically causes skin infections and sometimes pneumonia, endocarditis, and osteomyelitis. Some strains can even cause gastroenteritis, scalded skin syndrome, and toxic shock syndrome. And let's not forget about the atypical bacteria. So not all bacteria are able to be stained using gram staining. Those bacteria are classified as atypical. For example, bacteria that do not have a cell wall are not able to be stained by gram stain. Mycoplasma is a genus of bacteria that lack a cell wall around their cell membrane. Without a cell wall, they are unaffected by many of the common antibiotics such as penicillin or other beta-lactam antibiotics that target the cell wall. Next, let's look at the classifications of the domain Archaea. The domain Archaea has 5 phylums: the Crinarchaeota, the Euryarchaeota, the Corarchaeota, the Thuamarchaeota, and the Nanoarchaeota. First, let's look at the Euryarchaeotas. This is a Pyrococcus furiosus. This phylum of Archaea includes methanogens, halophiles, thermophilic, and sulfur-reducing archaea. Euryarchaeota, the first type of Euryarchaeota archaea we'll talk about are the methanogens. The methanogens are a group of archaea that are able to produce methane gas. More than 2 billion tons of methane gas is released by these methanogens each year. 1/3 of the methane gas release actually comes from cows. Interestingly, millions of tons of methane gas is released from methanogenic archaea in the GI tract of cows each year. Methanogens are also anaerobes. The definition of an anaerobe is that they do not require oxygen. These methanogens, however, are not only not dependent upon the presence of oxygen, but they will actually be killed in the presence of oxygen. Many of these methanogens live in mud at the bottom of lakes and swamps because it lacks oxygen. They are also found in the gut of some herbivores like our cows and some humans. Also, methanogens can be found in dead and decaying matter. The Euryarchaeota also contain halophiles. The halophiles require large concentrations of salt or sodium chloride in order to survive. Halo means salt, like the halogens on the periodic table, and phil means loving. These halophiles are aerobic, meaning that they require oxygen gas in order to metabolize. They are basophilic. They are found in high pH environments. And they're often found in places like the Great Salt Lakes in Utah and the Dead Sea. Next, let's talk about the Crinarchaeotas. The Crinarchaeotas. The name Crinarchaeota, it means scalloped archaea. They are often irregular or somewhat scallop-shaped. The phylum includes acidophiles, hyperthermophiles, hyperthermophilic sulfur metabolizing archaea, and we've actually found they're pretty morphologically diverse. Here's an example of a Crinarchaeota. This is the Sulfolobus sulfataricus. Let me try that again. The Sulfolobus sulfataricus. Try to say that 5 times fast. It was first isolated and discovered in the Solfatara volcano. So this guy's awesome because he was discovered in a volcano. Not like they actually have a gender or anything, but so this Sulfolobus sulfaticaris, I'm getting closer, lives in sulfuric acid hot springs at temperatures of around 80 degrees Celsius and a very acidic pH of around 2 to 4. Methanopyrus candelari holds the heat record for being able to survive temperatures of up to 122 degrees Celsius. Now remember, the boiling point of water is 100 degrees Celsius. Let's next look at the nanoarchaeota. The nanoarchaeota get their name from a Greek word meaning old dwarf. They are hyperthermophiles that have their optimal growth at 90 degrees Celsius. They only grow and divide on the surface of another archaea, which is the Ignococcus. Nanoarchaeota are the smallest organisms that we know of. Nanoarchaeota are the smallest living cells ever discovered, measuring only 0.4 micrometers. Nanoarchaeota also have the smallest genome of any living organism we know of, measuring only 480 kilobases. Chorarchaeota. Chorarchaeota. This phylum gets its name from the Greek word meaning young man or woman. They are hyperthermophiles, and their pH is more around neutral, about pH 7. They are found in high-temperature hydrothermal environments. These bacteria were discovered in Yellowstone National Park. Thumararchaeota. The Thumararchaeota gets its name from the Greek word that means wonder. This odd name for this phylum came about due to the sequencing of the C. symposium genome in 2008. The C. symbiosum genome was found to be so unique from the other known archaea that it created its own phylum. All species belonging to this phylum discovered so far are chemolithoautotrophic ammonia oxidizers. They play important roles in the nitrogen cycle and the carbon cycle. They also have type 1 topoisomerase that was previously only thought to exist in eukaryotic cells. So, as you may have heard, some of these names are quite tongue twisters. So you might be wondering, where do some of these names come from? So I would like to spend a moment and discuss nomenclature. Nomenclature is the fancy word for name. So there are a multitude of life forms on our planet. New species are being discovered all of the time. The convenience of categorization is that it assists us in our communication. Species are assigned a scientific name. Also, they can be called, their scientific name can also be referred to as their binomial name or their Latin name, according to their classification. These names are composed of two words. The first word is the genus, and the second word is the specific epithet. The genus and the epithet together define the species. Sometimes bacteria are named after people. Escherichia coli was named after Theodore Escherich, who first identified the common bacteria in stool. Neisseria gonorrhea was named after Albert Neisser, who first discovered it. Sometimes these archaea or bacteria are named according to their appearance. For example, Staphylococcus aureus received its name from the Latin words staphylo, which means clusters, and round, due to its round shape. It somewhat resembles a cluster of grapes. Some names are derived from the Latin terms that relate to their shape or physical appearance. For example, Vibrio cholera was named Vibrio, meaning comma-shaped, because it kind of looks like a comma. Nomenclature can also be based on physiological or chemical attributes. For example, Saccharomyces cerevisiae got its name because it was first observed in yeast cells, which were commonly used for fermentation. The name was assigned by Theodor Schwann in 1837. The word Saccharomyces comes from the Latin word meaning saccaro, meaning sugar, and myce, which means fungus. Yeast is a single-celled fungus that converts sugars into alcohol through a process called alcoholic fermentation. The word cerevisiae is derived from the Latin word cerveza, or cerevisia, which means beer. One last word on scientific names: how to write a scientific name. The genus should always be capitalized and italicized, whereas the epithet name is not capitalized but is italicized. The genus name can either be written out fully, as in Escherichia coli, or it may be abbreviated as E. coli. Alternatively, the scientific name may be underlined instead of italicized. Growth and nutrition for happy, healthy cells. What does growth in microbiology really mean? When we talk about human growth, we mean that the individual is bigger. In microbiology, growth refers to an increase in size or number of cells in the microbe population. In microbiology, a population of microbes grown on an agar plate is called a colony. So another way to discuss growth in microbiology is to say that the size of the colonies and/or number of colonies has increased. When bacteria cells are allowed to grow on an agar plate, one single bacteria cell will reproduce using binary fission, and it will give rise to a colony or genetically identical bacteria cells that were all generated from one single parent cell. Prokaryotic organisms reproduce rapidly. They reproduce using a process called binary fission. Binary fission is an asexual process. It creates genetic clones, and there's no genetic recombination. It is simpler than mitosis, which occurs in somatic cells, or your non-sex cells, non-gametes, of eukaryotic organisms. Binary fission requires less energy than mitosis. In a nutshell, binary fission occurs through the following process. The bacterium, before binary fission, has the DNA tightly coiled. The DNA of the bacterium is replicated. The DNA is pulled into separate poles of the bacterium as it increases size to prepare for splitting. The growth of a new cell wall begins, and the separation of the bacterium ensues. The new cell wall fully develops, resulting in the complete split of the bacterium. The new daughter cells have tightly coiled DNA, and they have ribosomes and plasmids. Prokaryotic cells reproduce using this binary fission. The interval of time between successive binary fissions of a cell or population of cells is known as the generation time or doubling time. Under optimal conditions, some species have a very fast generation time. For others, it can be much slower. For example, Staphylococcus aureus will have a generation time of 30 minutes. Mycobacterium tuberculosis has a generation doubling time of 15 hours. And Troponium palladium, Troponema pallidum spirochete, which causes syphilis, has a generation doubling time of 33 hours. When we analyze bacterial growth, it is convenient to create a graph of the number of bacteria versus time. This type of graph is called a growth curve. Analyzing these growth curves reveals distinct phases of bacterial growth. The lag phase of the growth curve is generated from the bacteria adjusting to their new environment. The cells will not undergo binary fission until they have adapted to their new habitat. The log or exponential phase of the growth curve reveals the active growth of bacteria in the population. Next, we have the stationary phase. The number of individual cells in the culture will continue to increase until the resources that they need to survive begin to dwindle. The bacteria begin to run out of space and nutrients, or when toxins are built up in the culture. The result is stationary phase, where zero growth occurs. In the stationary phase, the number of bacteria in the population will remain fairly constant due to the fact that the number of cells being created will be roughly equal to the number of cells dying in the culture. Then we get to the death phase. After some time, the depletion of nutrition and space and the buildup of toxins in the culture will cause more cells to die than cells being produced. This is the death phase of the growth curve. Endospores are dormant structures that can endure times of environmental stress. A few gram-positive bacterial species, such as the soil-dwelling bacterium of the genera Bacillus and Clostridium, produce endospores in times of stress. When environmental conditions are good, bacterial cells grow, mature, and reproduce as vegetative cells. However, when conditions for growth become strained, these bacteria are able to survive harsh conditions through spore formation or sporulation. Sporulation begins when the bacteria chromosome replicates. Then, an asymmetric cell division using binary fission will occur. The smaller cell will become the spore. The larger cell is the mother cell. The mother cell engulfs the spore. The spore forms a cortex and then a protective coat. The cell will stay in this endospore state until environmental conditions improve. When conditions improve, the mother cell expels the endospore and undergoes germination, which allows it to return to its normal vegetative state. What are the physical conditions needed for growth? Cells have temperature requirements. Different microbes have different temperature tolerances and requirements. Microbes will be able to grow at least to some extent when the temperature is within a particular range. However, microbes do have an optimal temperature that allows them to grow or reproduce more rapidly. This temperature is known as the optimal growth temperature. Psychrophiles require temperatures that are below 20 degrees Celsius. That is very, very cold. The freezing point of water is 0 degrees Celsius, and these psychrophiles are actually fine at that temperature. Mesophiles grow best at temperatures ranging between 20 degrees Celsius and 40 degrees Celsius. That's about our range that we like to enjoy. Thermophiles require temperatures above 45 degrees Celsius. And hyperthermophiles like it very hot. They want it to be at least 80 degrees Celsius or above. That means that these organisms are able to survive in boiling water temperatures, and the boiling point of water is 100 degrees Celsius. There are also pH requirements for growth. pH is the amount of hydrogen ions, protons, or H+, or hydroxyl ions, which is OH-, that are free in a solution. The reason we refer to the acidity or basicity or alkalinity of a solution as the pH value is because the pH value is the potential of hydrogen. The value of pH is calculated as the negative log of the molar concentration of hydrogen ions in the solution. Living organisms are sensitive to pH because the amount of hydrogen ions or hydroxyl ions will interfere with their normal chemical reactions inside the cell. Microbes can be categorized according to their preference and tolerance of pH. Acidophiles are able to grow in acidic environments with a pH as low as 0.0. Neutrophiles grow best in neutral environments with a pH around 7.0. Alkalophiles grow best in alkaline environments or basic environments with a pH as high as 11.5. We also must consider the physical effects of water on growth. Water is necessary to sustain life. Water is known as the universal solvent because it is able to dissolve more substances than any solvent known. Microorganisms need water to dissolve substances in the cell and to participate in metabolic processes. Enzymes and nutrients in metabolic processes. Also, enzymes and nutrients are needed to act as a reactant in many metabolic reactions. In nature, substances, including water, diffuse from areas of high concentration to low concentration. Cells are surrounded by a semi-permeable plasma membrane that allows only solutes to travel across. Only certain solutes to travel across. This selective permeability will create osmotic pressure due to the amount of dissolved substances or solutes in the extracellular fluid versus the intracellular fluid. When there are more dissolved substances in the surrounding fluid than there are inside of the microorganism, we call this a hypertonic environment. In a hypertonic environment, water molecules will diffuse out of the microorganism, causing the microbe to undergo crenation or shrinking, which leads to cell death. When there are less dissolved substances in the surrounding fluid than there are inside of the microorganism, we call this a hypotonic environment. In a hypotonic environment, water molecules will diffuse into the microorganism, causing the microbe to undergo lysis or bursting, and, of course, death. Different microorganisms have different preferences and requirements of osmotic pressure. Microbes that do not have a cell wall can experience a phenomenon known as osmotic shock, in which crenation or lysis can cause death due to the osmotic pressure. Microorganisms that have a cell wall are more resistant against osmotic pressure, yet they can still experience osmotic stress under certain conditions. So, we can classify microbes based on their preferences and tolerances for osmotic pressure. These are our salt lovers or halophiles. Obligate halophiles require high osmotic pressure, such as the osmotic pressure provided by salt water. Facultative halophiles do not require high osmotic pressure, but they are able to tolerate the high osmotic pressure created by the salt water. Let's talk nutritional needs. All living organisms are made up of around 96% or so of only 4 common elements, which is carbon, oxygen, nitrogen, and hydrogen. For this reason, the most important elements needed for microbial growth are carbon, oxygen, nitrogen, and hydrogen, just as you guessed. In order for microbes to be able to use these elements, they must be able to metabolize compounds containing these nutrients. Oxygen is Oxygen is not required for all organisms. In fact, in its molecular form, O2 gas, it can be toxic to some organisms. Organisms that do require oxygen obtain it from the air or from the water. Obligate aerobes are prokaryotes that require oxygen for their metabolism because they undergo aerobic cellular respiration. Humans are an example of an obligate aerobe. If you don't believe me, just see how long you can go with holding your breath. Humans absolutely depend on the presence of oxygen. Prokaryotes that do not need oxygen or cannot be exposed to oxygen only undergo anaerobic metabolism or anaerobic cellular respiration. These are classified as obligate anaerobes. Some species of obligate anaerobes cannot tolerate oxygen and will die if exposed. Facultative anaerobes are prokaryotes that exhibit metabolic flexibility. These microbes have metabolic pathways for when oxygen is present and another set of metabolic pathways when oxygen is unavailable. Aerotolerant anaerobes prefer to live in the absence of oxygen, but they can tolerate being in the presence of oxygen without dying. These organisms are able to tolerate oxygen because they contain vital enzymes that detoxify dangerous oxygen-containing compounds that would have otherwise had toxic effects on the cell. Microaerophiles are microbes that require low levels of oxygen to thrive. Hydrogen is needed by the cells as a key component of macromolecules. Hydrogen functions in the transfer of energy. Hydrogen is simply one electron and one proton. Hydrogen can donate its electron and become a hydrogen ion. In autotrophs, hydrogen is required for oxidation reduction. Organisms must obtain energy or hydrogen from the environment. Nitrogen is also needed by organisms. Many macromolecules contain nitrogen, including proteins, ATP, and the nitrogenous bases on DNA and RNA. Some microorganisms are able to undergo nitrogen fixation, which allows them to obtain nitrogen from the air, which is nitrogen gas, and then they convert it into ammonia. Others obtain nitrogen through the process of others obtain nitrogen through processing nitrates or even processing ammonia and urea. Sulfur is used by microorganisms for protein synthesis, cellular respiration, and photosynthesis. Organisms obtain sulfur from sulfur-containing amino acids, methionine, and cysteine, as well as sulfates and sulfides. Phosphorus is also needed in cells for the production of some nucleic acids and phospholipids, as well as other very important molecules such as ATP. Cell culture. Microbiologists collect a small sample of microbes from a given source. We call this small sample an inoculum and place them into a medium that contains the nutrients needed for life. This creates a cell culture. The medium can be in a liquid form called broth, or in a somewhat solid, gelatinous form called agar. Microbial growth using an agar plate will yield individual colonies of cells that hopefully have been derived from one single parent cell. Microorganisms are everywhere, on our skin, on surfaces, even in the air. Because of this, it's important to protect cell cultures from contamination. Microbiologists are usually intending to grow what's called actinic cultures. An actinic culture will consist of a colony of cells generated from a single parent cell or progenitor cell or colony forming unit or CFU. In order to produce such a cell culture, efforts must be taken to sterilize any substance, media, loops, glassware, etc. that may come into contact with the culture. Sterile techniques have been developed to reduce the risk of contamination. Here is an example of the streak plate method. In this technique, a sterile inoculation loop is used to spread an inoculum across the surface of an agar plate in a petri dish. The dish or plate will develop colonies following a time of incubation. After given time, in order for growth to occur, a successfully streaked plate will show progressively thinning colonies with completely isolated colonies. There is also the pour plate technique. In this technique, colonies are isolated through a series of dilutions. These dilutions are called serial dilutions. The final dilution is then added to warm agar in a petri dish. The dish will produce colonies after an incubation period. In this technique, colonies are isolated. We can see that aerobic bacteria will want to migrate towards the top of the agar so that they are more exposed to oxygen. More anaerobic bacteria will be deeper within the agar. Agar is the most widely used culture media. It has no nutrients, but it is often added often added to nutrient broth to form nutrient-rich agar or nutrient agar. It consists of a complex polysaccharide that has a low melting point and is easy to work with. Warm agar can be poured into petri dishes to form petri plates or agar plates, or poured into test tubes, and you can even keep those test tubes as a slant, at a slanted angle as it solidifies, so that you can get slant tubes or simply slants. And there are many, many other types of media as well. There's nutritive media, which is defined as any media that supports the growth of a wide range of microorganisms. This is often what we're using in the lab. Differential media is manufactured to undergo a visible change depending on the types of colonies that you want to produce. This media assists microbiologists in identifying more than one microbial species which may be present in a sample. Reducing medium is used when cultures of anaerobes are necessary because it contains compounds that remove free oxygen radicals from the solution. We also have selective media. Selective media contains ingredients that will inhibit the growth of certain molecules while allowing the growth of other molecules. A selective medium contains ingredients to inhibit the growth of certain microbes in a mixture while allowing the growth of other microbes. Differential medium are those that are able to distinguish microorganisms from one another based on growth characteristics that are visibly evident when grown on a specific media type. Organisms with differing growth characteristics typically show differences in growth media when placed on differential media. Examples include blood agar, eosin methylene blue or EMB agar, mannitol salt agar, and McConkey agar. EMB agar is considered a selective media for gram-positive cells. It is considered a differential media with respect to lactose fermentation. It contains eosin and methylene blue. EMB agar plates inhibit gram-positive organisms from growing. So this media is selective for gram-negative species. Lactose fermenting organisms such as E. coli produce a black precipitate on EMB agar. These colonies will be either black or possess dark centers with transparent, colorless peripheries. Non-lactose fermenters such as Proteus, Salmonella, or Shigella appear pink or uncolored. Blood agar is 5% sheep's blood mixed with normal TSA plates. It is a nutritive medium with differential properties with respect to hemolysis. Hemolysis is the destruction of erythrocytes, which are the red blood cells. The degree to which the erythrocytes are destroyed can be recognized by the effects of the hemolytic enzymes on the cells in the medium. Here's an example of hemolysis on blood agar. The complete breakdown of red blood cells is termed beta hemolysis and is recognized by clearing around the colonies, creating the hemolysin. Partial destruction of red blood cells leads to a greenish-brown color and is termed alpha hemolysis. Gamma hemolysis is usually the term applied to growth on blood agar that causes no damage to the red blood cells and no change in the medium. How do we preserve our cultures? Refrigeration will do for a short period of time. This cold temperature slows the growth of most microbial species without causing death. This is good for short-term storage, about 3 to 7 days. Deep freezing between -°C and -95°C, or long-term storage requires colder temperatures. Lyophilization is the process of freeze-drying the cell culture. Here, the cell culture is first frozen, and then the water content of the culture is removed using a vacuum. This is good for long-term storage.